welcome. I am today going to talk about whether or not we should genetically modify our human genome, the human genome. We cannot do this today, although there was a very, very nerdy April Fool's joke circulating from a biotech company that said, check out our new service, we can make your babies glow. But at the very end of the message, it did thankfully say, this is an April Fool's joke, we cannot do this, and we would not do this. But it's sort of in the scientific zeitgeist right now, to be sure. So what kicked this off? In March of last year, in sort of rapid-fire fashion, in March, all three of these were in March, you had three different groups come out with statements stating that we should not modify the human germline. Now, why did this happen all in very rapid succession? It happened because there was a rumor going around the scientific community about a number of groups in China who had, in fact, used a new technology of genome editing, which I'll talk about in a minute, to modify human embryos. And this rumor had been going around for a long time. It became more than a rumor when people started reviewing papers about this. And so in very rapid succession, a number of groups came out and said, we should not do this. Okay? These are March 12th down to March 19th. So then it happened. This was, I'd like to point out, submitted on March 30th and accepted on April 1st. If you know anything about how academic paper reviews work, this is impossible. So very, very rapid acceptance, very high level of suspicion about the level of review it actually received. But nonetheless, those three statements came out in March. April 1st, not an April Fool's Day joke. The first group in China published that they had, in fact, used these new technologies to modify human embryos. Now, the embryos that they used were triploid, meaning they had a whole extra set of chromosomes. So we got a set of chromosomes from mom, a set of chromosomes from dad, so we have two sets. These embryos had three sets, never ever could have become fetuses, never could have become babies. And they used these embryos specifically for that purpose. They were trying to walk an ethical line, but still do this work in human embryos. So this came out. We know that there are still a number of other groups in China who have done similar work, not necessarily in triploid embryos, but that work remains to come out. So first, before I get back to those issues, I'm going to take a little step back and talk about the kinds of modification we do today and are contemplating. But first, I'd just like to take a quick poll. By a show of hands, how many people think we should be allowed to genetically modify human embryos? And how many people think we should not be allowed to modify human embryos? All right, so a little bit more on the yes side.
Undecided?
How many people don't know? Yeah. Totally fair. Okay. Thank you very much for the corrective. So, what do we do currently? There are different ways of modifying DNA. There are also different sorts of DNA we can modify. So we can modify somatic DNA. That is DNA in the cells in your body, like in your cheek cells. That's not going to go anywhere. That's going to be a modification in you and in no one else. We can modify the germline. That's sperm and eggs. If we modify the germline, that could be passed on to the next generation. Right? Sperm and eggs create embryos, create the next generation. If we modify the sperm and eggs, there's the potential to create a modified embryo and therefore potentially human. There's also the question of mitochondrial DNA, which is sort of a whole other talk, but it's a really interesting talk, but I'm not going to give it here. But mitochondrial DNA is a different kind of DNA that we have inside our mitochondria, inside all of our cells, that doesn't contribute to the kinds of traits we normally think about when we think about what makes you, you, and me, but rather have to do with metabolism for the most part. Now, what are the different kinds of techniques that we can use to modify that DNA? We can use gene transfer, or you might know it as gene therapy, right? So we can modify the genes in a population of cells and put those cells into an individual to try to affect a cure. For example, of sickle cell anemia, you can take the blood cells out, modify that DNA, put those cells back in an attempt to treat the disease in that individual in front of you. We can do gene editing. That is genome editing, again, the gene editing of the sort that I will talk about in just a minute. And then there's also this question of mitochondrial transfer, which again is another talk. But for any of the geneticists in the room, I wanted to make sure you knew what that was. So, gene transfer. Gene transfer technology, or gene therapy, is a somatic treatment. You're delivering a gene that you think is going to help the individual to treat the patient in front of you. Historically, we've done gene therapy for single gene defects, but that is expanding, meaning diseases that are caused by one gene breaking. And the first clinical trial actually began in the 1990s. So we've been doing this for a long time. That first trial was a bit problematic, but we now have done about 450 trials in the US. So this is no longer a new technology. It's still cutting edge, it's still rare, but it's something that we have a fair amount of experience with now and have regulatory structures in place, policies in place to oversee that. Those policies include primarily the Recombinant DNA Advisory Committee, or the RAC. Side note, scientists are really bad at naming things sometimes. Naming an oversight body the RAC, that sounds like something you are being subjected to that is tremendously painful, maybe not the best idea or the best message to send, but nonetheless. The RAC was created in 1974 to regulate brand new recombinant DNA technology, so the ability to cut and paste DNA, like you cut and paste words in a text editing program. They meet 4 times a year. They used to oversee recombinant DNA work. They now primarily oversee gene therapy trials, or gene transfer trials. It is also the case that the RAC already has in their policies that they will not at present entertain proposals for germline alterations, but will consider proposals involving somatic cell gene transfer. So the RAC has already said, we're not looking at your germline gene editing proposals. So what's the deal with these new genome editing technologies? Why are they so sort of hot and sexy right now? Well, in the past, genetic modification has been really inefficient. So, I mean, we've been breeding animals for a very, very long time, right? So you can do breeding of livestock. You can take a bull, for example, that has a trait that you really like and use that bull to breed and try to get that trait into a whole line of cattle. Another way that, but you can't really control that, right? You have to wait for a trait you like to pop up in the natural population. So that takes a long time. You could also, and we do this for small organisms like yeast and small animals, you can do something that mutates, puts in a lot of mutations, right? Like you can expose the cells to radiation. And it creates lots and lots of mutations, but then you have to sort through all of those and figure out which ones are the ones you want, and which ones are the ones you want to try to pass on. But if you're talking about genetic modification for a therapy in humans, this is never going to work, right? We're not going to irradiate people and hope we get a mutation in the thing that helps them, right? That's never going to fly. So what we want is this, right? We want to be able to very specifically modify a gene of interest. And that's what these new gene editing technologies have allowed us to do. So The different types of technologies are zinc finger nucleases, transcription activator-like effector nucleases, which everyone calls TALENs for obvious reasons, and clustered regularly interspaced short palindromic repeats, CRISPR. Zinc finger ZFNs were discovered in 1996 and were used in flies to do genetic modification in 2002. But CRISPR, which has by far become the favorite for many reasons, including that it's it's easier and cheaper. This is the one that I'm really focused on for the most of the rest of my talk. Actually, I'll go back here. So what is that? What is CRISPR doing? What CRISPR is really doing, and all of these other technologies as well, is breaking the DNA in a very specific place and then repairing it in a way that you want it to be repaired. Either by creating a deletion so that you're actually breaking a gene that you want to break, or by putting in a particular gene or extra piece of DNA that you want. Okay, so you can very specifically insert DNA or break DNA. So what are the uses of this? So you can use this for somatic or germline therapies. We actually already have 2 examples of somatic therapy, one in HIV and one a little girl who had a cancer that was treatment resistant, and they used gene editing, and so far she is doing well. We have not done germline therapy as yet. But what are the uses of this? So you can do reverse genetics, which I described earlier. When you want to induce mutations, you can put a bunch of mutations in there and then kind of sort through them and try to figure out what was broken by each of those, right? The reverse of that is breaking something specifically and seeing what happened, right? So rather than going from the phenotype down to the gene, you're going the other way around. You're breaking the gene and looking to see what changes it causes in the organism. Gene therapy, like as I've just mentioned, and then potentially human germline genetic modification. Because again, if you can very specifically cut and paste and insert genes, you could do this in sperm as well. Or you could try to do this in human embryos, which would create a generation of individuals that was genetically modified in a specific way. This is also powerful right now because of its convergence with pluripotent stem cells. So these are a kind of stem cell, not necessarily embryonic stem cells are pluripotent, but we can also take adult cells and make them pluripotent and then make changes in those. So again, these technologies are much more specific than anything we've been able to do before, but there still are occasionally some off-target events or off-target effects. So you mean to just cause a mutation there, but there's another place in the genome that looks a lot like that, and it gets changed too. Those turn out to be really hard to detect, which is one of the problems. So what's the oversight here? Here, the oversight would be the RAC, which, as I already told you, has said they are not going to look at germline proposals. And the FDA, we would also, anyone wanting to do this would also need an investigational new drug license from the FDA to attempt this in humans. Internationally, about 40 countries have relevant policy to the question of human germline genetic modification, and they all ban it. These policies fall under a range of kinds of policies, including stem cell science, reproductive medicine, and gene therapy. And in many of these policies, there is this important distinction between germline and mitochondrial DNA. I'm going to leave that to the side. We can talk about it in the Q&A if you want. So this takes us back to these 3 statements that came out saying, do not do it. So more specifically, what did they say? The Baltimore et al. Paper, this is a group of academic researchers and ethicists. They strongly discouraged attempts globally. Again, remember, this is when the rumors were still rolling around and before that paper was published. So they're saying, we know you're out there and please don't do it. Create expert forums for public education and discussion. Encourage and support basic research, non-reproductive research in CRISPR-Cas9, and convene an Asilomar-like meeting for human germline genetic modification. Asilomar is the meeting that led to the creation of the RAC. So they are calling for a meeting that would lead to some sort of governance model for this science as well. The Lamphere et al. Statement, this is a group of biotech executives. Very different set of interests and motivations. They said there's absolutely no reason to do this, it's not safe, it's not legal, and we need to talk about it internationally. The primary professional body for stem cell science also called for a moratorium on attempts to do this editing of the human germline. And the NIH said, we're not going to fund it either. So a lot of doors seemed to be closing. But it's important to note that this isn't the first time we've had this discussion. We've actually been talking about human germline genetic modification for a very long time, not only in this country, but internationally. In fact, this report, you can see, a report on the social and ethical issues of genetic engineering with human beings, 1982. This report was begun under Jimmy Carter and was delivered to Ronald Reagan. These are not new conversations we're having, right? We've been talking about this for a very long time, even in the media. You look at these headlines. Gene technique could shape future generations. Ethicists wary over new gene techniques' consequences. That's Gina Colada in 1994. I mean, she still works at the New York Times. She could put that headline in an article today and it would be just as relevant. So what were the major concerns last time around? Safety, to be sure, and I'll come back to that one. Informed consent. How do you do informed consent for technology that's going to modify an individual who doesn't exist yet? Right to an open future. You are making specific genetic changes in an individual who again, doesn't exist yet, and you're making certain decisions about what that person's life is going to look like through the kinds of modifications you make. And are the kinds of modifications you think are important to make, would they be the same kind that that individual would think would be important to make? Commodification. This is always couched or described as being about designer babies, right? How much control are we taking over our kids and over the next generation? Questions of distributive justice. Who's going to have access to this? Say this does turn out, we do decide to do it, and it turns out to be a great thing, and we're able to avoid disease in many, many individuals. Who's going to get this, right? It is never, new technologies are never available to everybody, certainly at first. Sometimes they trickle down, many times they do not. So there are serious questions about distributive justice. And then there are questions about treatment versus enhancement. Again, if we're going to do this, for what things will it be permitted? To avoid very serious disease, maybe people can get on board with that. To change someone's eye color, feels a little bit different. To make someone more intelligent, right? People feels even or less uncomfortable to many people. The conclusion at that time was we should not do this. Although that was largely because it wasn't safe, right? I mean, you saw what the mutation, how we used to do induced mutations, right? Not safe at all. But now a lot has changed. CRISPR, first of all, is super easy and really inexpensive. Science is bigger, right? And the Asilomar meeting that led to the creation of the RAC, There were about 140 people there, and it was pretty much the entire scientific community that was doing that work. They could get them all in a room for a few days. That is not true anymore. Science is international, and it's a very different landscape. Also, the rest of science has moved on, right? We've sequenced the whole human genome. In fact, we can sequence a human genome for $1,000 now. It cost us $2.7 billion to do it the first time. And it's now $1,000. That's a big change, right? We have a much, much better understanding, or at least much better access to the genome than we did in 1982 when that report came out. And there are also advocates for this. There are people who think, in fact, we have a responsibility if we can avoid disease in the next generation to do it. And then the last issue, and this has been sort of the specter that's been floating around and that motivated part of my title, is that in stem cell science, we have, over the last 10 years, been having a terrible, terrible problem with clinics internationally who are more than willing to take your money to inject you with who knows what in the name of stem cell science. And the field is frankly terrified that this technology, because it is cheap and it is easy and it is specific, is going to start offering to do that too. There is a very deep concern that rogue clinics that are more interested perhaps in their bottom line than in the health of individuals, are going to take up this technology and start offering it. And there are going to be germline genetic modifications before anyone thinks it should be done. And that would be terrible, not only potentially for the individuals who underwent that, but for the field generally. So all of this has spun off a ton of conversations, deliberations, debates about what to do, about how to think about now that safety, we can't quite hide behind safety anymore and say, well, the technology isn't there yet, we simply can't do it. We're actually going to have to start engaging some of the more difficult questions about modifying the next generation. So these are some of the groups that have been working on this. The Hingston group, full disclosure, I run this group. I'm going to talk a little bit about our consensus process and what we produced. And then I'm also going to talk a little bit about what the National Academies of Science and Medicine have done and are doing. So the Hingston group, we are a group of international, interdisciplinary folks who are interested in ethics and policy issues in stem cell science. We held a meeting in September of 2015. It was the 5th meeting of this sort that we've had, not around this topic. They've always been different topics. But we met to talk about how do we think about and what should we do about human germline genetic modification. What we came up with is categorized under sort of 3 basic headings. The first is the value of basic research. This is non-clinical research in the lab. And I want to say that these are all direct quotes, but they're not the whole thing, and I can assure you we absolutely agonized over the language. So please, if you're interested, go and read the whole thing. So with regard to the value of basic research, our group did in fact think that genome editing has tremendous value as a tool to address fundamental questions in biology, right? Because of reverse genetics, because of the ability to say, you know, I want to break the cystic fibrosis gene in the very specific way that it is broken in this individual and figure out what happens at the cellular level, right? Basic scientific questions that may have clinical implications, but are all happening there on the lab bench. And we said that it's our conviction that concerns about reproductive uses of these technologies should not halt or hamper the application to scientifically defensible basic research. We recommend that a detailed roadmap be produced to guide the development of standards of safety and efficacy. So we need to figure out, what do we need to know about this technology in non-humans its application in mice and rats, in monkeys. What do we need to know to make us, to give us the information we need to make a decision about whether it will ever be safe in humans? And not only what do we need to know, but what are the experiments we need to do to get there? We recommend that scientists carefully consider the category of embryo used. For example, triploid embryos as opposed to normal diploid embryos. And that all of this science should be made publicly available. Especially not only because the questions are much bigger than any individual research project, right? We're making a decision about what the national or global community should do in this area of science, so we should all share our data so we all know what's going on. The second category had to do with the prospect of human genome editing for clinical reproductive purposes. So should we permit human germline genetic modification? And we said, right now, absolutely not. We don't have enough data. We do not know enough about off-target events or the genome, frankly, to do this right now. We wanted to make a very clear statement to those rogue clinics that this is not something any legitimate scientist or clinician should be doing right now. We should have international and regional debate to make decisions about not the safety necessarily, but the ethical acceptability and permissibility of different potential uses. So, for example, use to avoid very serious disease that's present at birth versus use to enhance an individual in a particular way. Before moving forward with clinical applications, attention should be paid to how applications are prioritized and based on what criteria. If we're going to think about serious disease first, should that be serious disease present in the US or serious disease that affects the most people globally? How are we going to think about prioritizing various uses if we're going to permit this technology to roll out? Individual societies are going to have to make their own decisions, right? Different nations have different laws motivated by different histories, right? Germany has very restrictive laws when it comes to genetics and stem cell science for totally understandable reasons, right? There is going to be international variation, and that variation, so those conversations about the appropriateness of these technologies should happen in individual jurisdictions. There's important work to be done to identify and explore the issues that are common and divergent across science in the dish and science in sperm and eggs in the dish versus science, for example, in a pregnancy, right? Whether it makes sense in any particular case not to modify the sperm or eggs, but to modify, basically do gene therapy very, very early in a pregnancy. Oversight structures must be in place prior to any attempts to do this in human reproduction. We need to have an entire architecture of oversight in place before anyone does this. The third category, which is very close to my heart, is the importance of governance and meaningful engagement. In these decisions, I mean, this decision is different than a lot of the decisions we've made in science in the past that are about particular changes in particular people, right? We're talking about modifying our genome going forward. And I think that sort of raises the bar in terms of which and how many voices are involved in the discussion that lead to the decisions about whether or not we should do this, and if so, how, or for what kinds of conditions. We need to come up with engagement models, ways to engage the public that are meaningful, like not just, yeah, tell me what you think and then I'll go and do what I want to do, but are meaningful opportunities for engagement with the public, and that have a transparent way to get that engagement process into policymaking. Like, is it just a process that, you know, so everyone can think about how they want to think about it, or is it actually informing the policy decisions, and how that is happening? In considering policies, there should be a distinction between objections that are based on technical concerns and objections that reflect moral considerations, right? It's not safe yet is different than a concern about the sanctity of the human genome, or concerns about the commodification of babies. Scientific journal editors should support and promote high standards for papers that report this science. So that paper that I showed right at the beginning of the group from China, that we know was reviewed by multiple journals before it finally found a home and was published one day after being submitted. So journals are an important gatekeeper in academic science, and they should take that role very seriously when it comes to this science. I think in the interest of time, I'm going to move on to the National Academies process. Sorry, I did not know that. So this came from Jeff Kahn's slides. I did not realize that his were automated. I apologize. So the National Academies are also undergoing in the process of, or undergoing a process to deliberate human germline genetic modification. It was sort of kicked off with an international summit in December of last year. It was jointly hosted by the National Academies of Science and Medicine, the Royal Society from the UK, and the Chinese Academy of Sciences. They had a range of perspectives on science, applications, social and ethical implications, governance, present at the meeting, both presenting and in small discussion groups. And it resulted in a statement from the summit organizing committee. This is also, there's frankly a fair amount of overlap between this and the Hingston Group statement, but the piece that I want to bring up here is this need for an ongoing forum. So they said we should create an ongoing international forum to discuss potential clinical uses of genome editing and help inform decisions by national policymakers and others. This group should formulate recommendations and guidelines and promote coordination among nations. And they actually very rapidly took this up for themselves. So there is now ongoing the National NAS-NAM Consensus Study on human gene editing, scientific, medical, and ethical considerations. Their first meeting followed the end of the International Summit, and that was their first meeting. Their second meeting was in February. Their third meeting is at the end of this month in Paris, and they are to issue a report in, there it is. They're to issue their report in fall of this year. So about a year-long process, which is actually lightning fast for the National Academies. So stay tuned for that. I'll be very interested to see what they have to say. This is the entire, these are all the committee members. The one thing that I do want to point out is right here, Jeff Kahn, who is on our faculty at the Berman Institute, and Robin Lovell-Badge, who's a member of the steering committee of the Hingston Group. I would like to thank my funders for the Hingston Group work, and thank you very much for your attention. I am more than happy to take questions. I would actually love to take questions. Before I do that, though, I'll point out one other thing, which is, I said, we wrote at the end of, or as part of our Hingston Group statement, the importance of meaningful engagement with the public. And the National Academies called for an ongoing forum, which they're doing, but there is not yet any, what the National Academies is not doing is any kind of engagement themselves. I'm very much hoping to do work on this, and that's one of the reasons I would love to hear what your questions are and what you think we should do with this brand new technology. Thank you so very much. Oh, can I take another vote? Okay, now, how many people think we should do this? We should do human germline genetic modification. How many people think we shouldn't? And how many people are still unsure? We got a little bit of movement to yes. That is so interesting. That was not my intent. Okay, questions? There was a hand back there. Yes, sir.
Does the CRISPR process operate on a single cell, one cell in and one cell out at the end that then reproduces?
No, so CRISPR, well, I mean, it can be done that way, but generally CRISPR is done on a population of cells. And so, for example, in the case of the little girl, I think it was bone marrow that was treated with CRISPR and then put back. So it's done on a population of cells, but that is what makes it tricky, right? Because you can't sequence all of those cells to see if there are off-target events. It's hard to tell the difference between a natural mutation that just happens in cell reproduction versus a mutation you caused with your technology. So it's devilishly tricky. Even since the technology really hit the scenes in human cells, like 2 years ago, we've gotten much, not me personally, we, the scientific community, has gotten much better and more precise with CRISPR. There's still, it's just hard to assess. Once you get below a certain threshold of off-target mutations, it's hard to figure out if they're there at all. Yes?
What are the implications of this technology for genetic conditions such as trisomy 21, Down syndrome, and other similar events that take place almost immediately upon conception? What is it that can be done? Do the same ethical issues apply as to choosing the color of your child's hair or eyes, etc.?
So first of all, trisomy 21 is a little bit different because you're talking about a problem that happened when the egg was being made and the chromosomes didn't separate properly. So you have an entire, whole extra chromosome, right? Which CRISPR can't really address unless we can figure out, and people are working on this and have had some success, which of the genes on the extra chromosome 21 are causing the neurologic disability or the heart problems, for example. In which case, then, maybe CRISPR could be used. With regard to what about things that happen very early in reproduction? Well, that goes to this point that I made earlier about figuring out when in development it makes sense to intervene. When we were preparing for the Hingston group meeting, we had a research associate, Theo Schall, who put together this fantastic developmental timeline from sperm and egg up through, we went up through about 14 weeks gestation, I think, trying to figure out when we could intervene in that process to still induce germline changes or not, right? So how early does the fetus have sperm or eggs? And if you could intervene before that, you could intervene in that individual very early but not cause a germline mutation. Is that even possible, right? That turns out actually to be quite tricky because at that point in development, A, you might probably don't know you're pregnant, and B, it's really hard to test the DNA of that fetus at that point. But these, I think, are really important questions in figuring out, for any particular disease, when it makes sense to intervene. Right? It may actually make more sense to intervene in the fetus than it does to do modifications in sperm and eggs in advance. But it's also the case that for many conditions that we're worried about, it's not something that the parents have. Maybe it's something the parents carry, right? You need 2 copies of the gene to make the disease, and the parents only each have 1 copy, and so they don't know that they're carrying that. So they wouldn't know to do any kind of testing or modification in advance anyways. So in that case, you would be talking about, well, how early in fetal development can we figure out what's going on, and does it make sense to intervene then, or wait until the kid is born? Any other questions? Yes?
How seriously should one take the results that one gets? I'm sorry, thank you. How seriously should one take the results that you would get from an internet genome test?
So meaning you send in a spit sample and they send you test results. So it depends on the company. And it is certainly the case, I think it was OMB, the Office of Management and Budget, I want to say, did a sort of sting operation where they sent the same exact sample to 2 or 3 or 4 different genetic testing companies and got very different results back. So I would, grain of salt, maybe big grain of salt, but also check out the company very closely to figure out how legitimate they are. I mean, 23andMe is certainly sort of the 500-pound gorilla in this regard, and they do now have FDA approval for a small number of tests. And I think they are sort of trying to get right with the law, which they were not trying to do for quite some time. Yes?
Are there laws in place concerning these rogue labs? The justice system have any criminal prosecutorial caveats concerning them, or do they just go along their merry way?
Well, it depends on what country you're in. So here in the US, we actually have a fair number of them, but the FDA is now they've exerted their jurisdiction over these clinics and are trying to shut them all down. But many of them exist overseas. So Kobe Bryant went to Korea to get his knee injected with, again, goodness knows what. But, I mean, he's a role model, and he's gone and done it, right? And there are clinics in China and India and Indonesia, and we certainly in the US have no jurisdiction whatsoever over those. But people will crowdfund to take their very sick children to these places to have them treated. It's very concerning. Yes, thank you.
Hi. So I think this issue also leads to another issue of fraud within the research community, because I had heard that particular study from China that they had made a pluripotent stem cell from a differentiated cell. That actual study was found to be false because researchers were sending their papers to friends, or they were either creating fake emails so that they could review their own papers. But I think, and how does that tie into this field as well? Because are those studies actually real? Because I know there was a series of studies that they had investigated that indeed the science was actually not real, and that Dr. George Daly from Harvard actually investigated whether that particular study that the Chinese investigators had made a pluripotent stem cell from a human cell was actually not real.
So I think you're referring to the Woo-Suk Wong papers from the mid-2000s. So this was an investigator from South Korea, actually, who had said that he had done human research cloning, so somatic cell nuclear transfer in human cells, and it was a very, very big deal internationally and in his country. He was made the supreme scientist of Korea. He had his own postage stamp. He had free lifetime travel on Korea Air. And it did, in fact, turn out that the work was fraudulent. And that is absolutely a problem. I think that it may be slightly more of a problem in areas of science that are super sexy, right, that could really make your career. And I think we do need to think carefully about the incentives in science, especially in areas of really cutting-edge science that are getting a ton of media attention. But I would also say that there are bad actors in every field, and that includes science. And there are policies that we can have in place to try to guard against those. So, for example, after the Woo-Suk Wong situation, where he said one of the things that was quite fraudulent about his papers was he had figure, and I have this amazing figure in a different presentation, of a figure from his paper that has, I don't know, 36 different little pictures that are supposed to be independent experiments, and it turned out they were all of the same picture. They were just cut differently and turned and flipped. And now many journals use software to detect that kind of manipulation in PDFs. So changes were made to try to guard against that kind of thing, but again, there are bad actors in every field, and that includes science. Yes? Oh. Sorry, I will come to you next. Yes?
When I was a resident at the University of Pennsylvania in the early 1990s, I took care of a man in the ICU, a young guy, who died a horrible, horrible death after a gene therapy trial. For relatively young use. What changes and safeguards have been implemented since that time to prevent this kind of recurrence like that?
So I know at that time that there was a very serious investigation that took place at Penn. I don't know what changes happened in oversight at Penn. I wish I did Journal Club on this story when it happened, but I can't remember all of the details. But a big part of it, I mean, it wasn't fraud, it was lack of due diligence, right? There were sources that could have predicted that bad outcome that were not sought out. So honestly, I don't know the answer to your question in terms of either at University of Pennsylvania or nationally. I don't know that there were changes nationally, but I'm not 100% sure. Sorry, up here. Oh, and then I'll come back.
For those of us as we get older and start worrying about things like, say, Alzheimer's disease, now, do you think the time might ever come when, say, if we're in our 60s and 70s, that some of this stem cell modification or something like that could be introduced even at that elderly an age as we get older that would help combat some of these things?
Certainly a source of research. And it is absolutely the case that researchers are trying to detect disease like Alzheimer's earlier and earlier, so we can intervene earlier to avoid the damage that happens, because it's hard to repair the damage once it's already done. So ideally, we would like to avoid the damage from happening. So possibly.
Hi, I'd like to shift from old folks to young folks. I know there's some work going on now for women who have defective mitochondrial DNA, to have that DNA replaced by a healthy one, and so the child comes out with genes from 3 people instead of 2.
Yep.
Are there any guidelines being contemplated for how to deal with those situations?
It's like you're a plant. So this is that whole other talk I mentioned a couple of times. So it is the case that there is a technology that has been developed in mice and in monkeys and has been approved for use in the United Kingdom, but not yet here, called mitochondrial replacement therapy. I think it's a bad name because we don't have a therapy yet. I call it mitochondrial replacement IVF. And it is the case that it can be used for mitochondrial disease only. So you have your nuclear genome. Those are the 46 chromosomes. You got half from mom, half from dad in the nucleus. But then outside of the nucleus, in the cytoplasm, the rest of the cell, are a whole bunch of mitochondria. And they generate energy for your cells. And mitochondria, that has its own, a tiny little genome. So there are about 23,000 genes in your nuclear genome. There are 37 on the mitochondrial genome. It's tiny, but you have lots of them. You have one nucleus. You have lots of mitochondria, lots in each cell. And how many you have depends on whether it's a muscle cell and needs lots of energy or whether it's another kind of cell that doesn't need as much energy. And they replicate independent of your nuclear DNA. They're very, very sort of funny. And because they're funny, it's really hard to predict if you have a mitochondrial disease, what the mitochondrial disease in your kid is going to look like. because it depends on how many mitochondria are in that particular egg that goes on to make that child. Because mitochondria are only in eggs, not in sperm, and so they're passed from mother to child exclusively. So if you're a woman with mitochondrial disease, there's no good way for you to have a genetically related child that you know won't have disease. And these diseases can be terrible. They can be mild, and they can be devastating. And there's no way to predict what the case is going to be for you. So this potential treatment has been developed wherein you replace the bad mitochondria that you have with mitochondria from someone who has healthy mitochondria. And the debate is that genetic modification? You're swapping it, you're not messing with the DNA itself, right? You're taking mitochondria, you're actually taking a nucleus and putting it from an egg that has defective mitochondria into an egg that has healthy mitochondria. So you're not messing with the DNA, you're messing with where the bits of the cell go. But you do end up with a kid that would have 3 genetic parents. Nuclear DNA from mom, nuclear DNA from dad, mitochondrial DNA from healthy donor. And as I said, the UK actually changed their law to make this technology permissible in the UK. No one has done it yet. The US had the National Academies do a great big report. So the FDA had a hearing to talk about the science. They turfed the ethics to the National Academies, to a committee that was actually run by Jeff Kahn from the Berman Institute. Jeff Kahn and his committee came out with a report that said it should be permitted, but only to produce, for the time being, to produce male offspring. Why? Because they're not going to pass on mitochondria. So that change stops with them. So if there are any generational issues with doing this, we won't see them when in the early experiments. A day or two after that report, the FDA said, actually, we're not going to allow it. So for the time being in the US, it's not going to be permitted, but it will go forward in the UK. Yes?
So in terms of more like the biological aspects, it seems like some of the ones you mentioned already exist, but it will like bring them to the extreme. Like in particular, for example, the social justice.
Yep.
Like why would this be expected to be equal and to kind of be devoted to big problems that affect developing countries when now that's not happening? I mean, it's kind of what happens already with pharmaceutical companies. So yeah, that's any prospect that I would be encouraged somehow.
So with all new technologies, I mean, I work, as Alan mentioned, I work on a lot of different emerging biotechnologies, genetics, stem cell science, neuroscience, synthetic biology, and I think that there aren't actually very many new issues. It's just sort of a different arrangement of issues in the particular environment, and each new technology is sort of a new opportunity to get governance right. Because we haven't gotten it right yet, I don't think. We certainly haven't perfected it, and I don't think it's going to be very specific to each technology. So, and it gives us an opportunity to try to wrestle honestly with the issues again, right? I mean, the social justice issues, we have that conversation all the time. Have we solved it yet? No. It's really hard. But each time we come up with one of these new technologies and we have to make a decision, we can try again. You know, and different jurisdictions can try again. And I don't think that we're necessarily going to solve all of them this time, right? But I think we have a good shot at a couple of them. Not distributive justice, I'm sorry to say. But every time that policymakers are forced to think about those issues, and the public is forced to think about those issues, I think that's a good thing. I think it'll get us closer to an answer. Yes. Last question?
One more
Okay.
question?
Right in the middle. Sorry, Becky.
couple of areas you think shows promise now?
Do you mean,
You think the couple of areas?
oh, different areas that I work in?
Yeah, so that
Or
is promising now.
for genetic modification?
Yeah.
I think that we're going to see more somatic therapies using gene editing, to be sure. And I think that that has tremendous promise because we already know how to deliver these things into people who are sick. And this gives us a new technology that will allow us to make very specific modifications of the sort we want. And I think it's going to be very good for gene therapy.
Thank you very